Hello everybody, welcome to the webinar for analyzing image stream data in FCS Express. Today we're going to be talking about loading in and working with Amnis image stream data in FCS Express 7. My name is Sean Burke, I'm the Senior Product Manager at DeNovo Software, and I'll be guiding you through the webinar today. So one of the big enhancements to FCS Express that was made a few years ago is the ability to read the native DAF and CIF files that are produced by Amnis instruments. So what that allows you to do is analyze all of your Amnis data while maintaining the linking directly to the images that come from the data from the cytometer. And what we're going to do today is give you an overview of how to load up some of those data files in the software, how to begin analyzing them, how to work between your kind of standard flow cytometry style plots and the raw images associated with the data files, and also walk through a few examples of some of the reporting functionality that FCS Express provides to kind of take that analysis for your Amnis data to the next level. So a good place to start is to talk about some of the benefits of using FCS Express to report results from any of the Amnis instruments. So essentially when we're working in FCS Express, there's a lot of kind of key concepts to keep in mind. Essentially, when you're working with FCS Express, you can completely, batch export all of your plots and images to kind of achieve higher throughput. So if you have a lot of data files that came off the Amnis instruments, we can quickly crunch through them. I know one of the kind of things that's difficult to do in Ideas software is create histogram overlays. Of course, FCS Express makes it very simple to do with a drag and drop. The software can also be used to perform data transformations. Tisney, Visney, Spade, clustering can all be done with your Amnis image cytometry data. If you do work in some sort of regulatory environment and you need 21 Part 11 compliance for your analysis software, FCS Express does has some functionality and features that can help you meet that compliance in certain versions of our software. The other kind of important thing is, of course, FCS Express is going to load all of the images in and features into FCS Express. So you have really a lot of the functionality that you're working with in Ideas, and you're not just throwing away those images that you painstakingly acquired. Also, if you're working with cell cycle data, if you're working with 96 well plates, if you're going to be doing downstream analysis in things like Excel and Prism for regressions and IC50s, a lot of that can actually be done directly in the software. So a lot of this you're going to see through some of the examples today. But we are going to take a step back and talk about the file format and how to get some of that data into FCS Express when you're ready to analyze it. So the important point here is FCS Express has direct compatibility with Ideas derived DAF and CIF files. That means there's no need to export two FCS files from Ideas software in order to get your data files into FCS Express. So, one thing that can happen is if you're using the export to FCS file function in Ideas software, of course, you're throwing away all the images that you had put together in Ideas. So, we recommend just pulling in the DAF and CIF files. If you're using other software to kind of analyze the FCS files, keep in mind FCS Express is just going to allow you to load those in. Now, the general workflow is when you create your data files from Amnis software, you open up the RIF files and Ideas software. That essentially automatically generates a DAF and a CIF file. The DAF file contains all of the numeric data associated with your data files. The CIF file contains all of the images. And really what you need to do is just keep the DAF and CIF files together in the same folder, and then you can load those data files into FCS Express. And what FCS Express is going to do is it's going to pick up where Ideas leaves off for reporting of the data. So, if you need to create plots for publication, if you need to get statistics, if you need to do IC/EC50s, FCS Express is a great choice for doing this. However, FCS Express will not perform segmentation, it will not perform the image compensation that Ideas does, it won't perform any sort of adjustments to your images. Those are all things that should be done in Ideas software. So we're going to take a look at that in FCS Express itself, loading some of the data and the basics to get started. We're going to show you that with a specific data file. We're going to teach you about inserting plots and working with data grids, how to kind of visualize images, how to do backgating, and then we're going to come back and take a look at a few layouts and templates for analysis that we think are kind of good uses of using the software with Ideas data. So what I'm going to do here, we're going to open up FCS Express. And the first thing I want to show you is how to get some data into here in the data file formats. So if I pull over a Windows file browser, what you're going to see here is that I have a DAF file that was created from Ideas software. I have a CIF file that was created from Ideas software. They're both in the same folder. So what that means is I can load up that DAF file into FCS Express and it's going to come in with all of the associated images. So there's a few ways to do this. What I'm going to do is I'm going to open up the data list in FCS Express. I'm going to choose to add a data file to the data list. And what I'm going to do here is I'm going to ask SCS Express to go to this location. And really the important thing here is that if you're using or if you had been using some sort of file format previously, or it's the first time that you're opening up FCS Express Image Cytometry, what you're going to want to do is make sure in this files of type you choose Amnis DAF files. Once you do that, you'll be able to grab that DAF file, open it, and put it into the data list. And when you're working with any of these Amnis derived data sets, it works just like any other analysis in FCS Express. So if we want to get a plot inserted into our layout, I can simply drag and drop. I can choose a plot type of interest. A plot will be inserted, and now we're starting to look at our Amnis data and all of the parameters that are associated with it. So again, that's one way of getting the DAF files into FCS Express, add them to the data list. Of course, the other kind of nice trick or way that you can get data file into the software is if you have it in the file browser, you can just drag it and drop it into FCS Express. Same thing's going to happen. FCS Express, if you drag and drop, it's going to automatically put that data file in your data list. But again, just another way to get to the same functionality. So again, what we're going to do here now is create some gates on this data, and we're going to show you how to access the images. But keep in mind that everything that we're doing today for these DAF files works the same way as you would analyze any standard flow cytometry data in FCS Express. So if I need to create a gate, I can create a gate on a population. FCS Express is going to prompt me to give it a gate name. We'll leave it as gate 1. If we want to drill down into this gate, we can drag and drop the gate outside of the plot onto the layout. It will create a new plot, and then we can pick and choose from any other parameters we want to analyze. Now with Amnis Data, you probably already know there's lots of parameters in our data sets, right? There's a lot more that we can get out of an image cytometry data file rather than just standard intensity measurements like we would get for flow data. So one of the things you can do is you can use the search functionality in the parameter selection dialog here. So again, the way that we get to this is we click on the parameter. If we start typing a value, it's going to jump to whatever the search finds for that. So if I wanted to quickly find the intensity of the MC channel 11 parameter, I can do that, and you can see that that gets displayed on the plot very quickly and easily. So one thing that I should note about the Amnis data files is if your scaling doesn't look appropriate for some reason, in this case, things are kind of squished down here on the axes, that's okay. You can always come in and format these plots by double clicking on them. When you format them, you can change things like the X and Y axis range. You can change in the specific options, the resolution range. But a real quick and easy way to kind of get into this data is if you right click on a plot, if you choose magnify resolution, what you're going to find is that you can draw a little box around the region of interest, and what that's going to do is zoom in on that population of interest. So that way I get a kind of closer view of that data. The other thing we can do is if we don't want to visualize just density plots, if we wanted to come into our format tab and change to something like a contour plot, change to something like a color dot plot, we can do that and we can use whatever plot type we want. Additionally, of course, you can always insert from the Insert tab any sort of plot type that you want, histograms, densities, dot plots. Now, the one thing we haven't talked about yet is how to get information from our images with this data. So far we've just inserted some plots that are going to show us numeric values, density plots, color dot plots. But if we want to start visualizing the individual images associated with all these cells, what we do in FCS Express is choose Data Grid from the Insert tab, and we draw a data grid on the page. Now, what a data grid will show you is each row is a cell in your analysis or in your data file. So there's lots of cells here. And each column is a parameter of interest. And what you'll find with the data grids, whenever we work with image cytometry data, is that at the very end of the data grid, you will find all of the raw images. So you can see here that for our Amnis data set, we have the composite images, we have some DREC 5 staining, we have some bright field images, we have an NF kappa beta, we have another bright field channel, so we have access to all of these images. Now, what you may not want to do is visualize all of this numeric data, right? You probably just want to be looking at these images. So just like any other plot in FCS Express, you can double click or you can right click and format, and there'll be a set of formatting options that appear. Now, the first formatting option that appears for data grids is the parameters to display. Now, what that means is if I want to only choose certain items that I check in this box, they will be displayed in the data grid. So for instance, if I want to show the composite, the DREC 5, one of the bright field channels, the NF kappa beta, right, I can include to show these within that data grid. And that's how we're going to access all of the images that are here. And an additional functionality within this formatting dialog here is gates to display. I'm going to enable this, and you can see here in gates to display it says no gates, hide the gates column. But if I choose all gates, what it's going to do is put a little box next to any of the cells that fall within a particular gate. So in this case, we have a red box, which is associated with gate 1. Any of the events that fall within that gate get a red box next to it. Like cell number 11 here does not fall within that gate, so it does not get a red box next to it. So again, setting up these data grids, it's pretty simple. All you have to do is insert the data grid, right click, do a format. You can choose your parameters to display, you can choose your gates to display, and there's also options here for data image options. I'm going to show what this means. Essentially, if you want to keep your images centered in this grid at a certain size, the default is to do that. But if you would like to fill the grid cell, so if I choose this option in data image options, what will happen is however large I make this grid cell, whether I expand the columns or the rows, FCS Express is going to try and fill that grid cell with the image. When I do that, of course, if you set it back, and you resize these columns, it will go back to the original view. So, just some things here to make working with these data grids a little bit easier and simple. Now, what I'm going to do in this layout, I'm going to insert a gate view so we can see our gating hierarchy and kind of keep track of that as we work through a little bit more of the functionality here. And I'm also going to create some additional gates here. So I'm going to call this gate, which is our MC channel 11 negatives, it's going to be called gate 2. We'll create one up here for our MC channel 11 positives. We will make this a green gate, and this will be called gate 3. And what you're going to find in that data grid is, again, we're visualizing the cells that fall within these gates. So cell number 2, it's positive for NC11. It falls within that green gate. It also falls within this gate number 1, whereas cell number 1 here looks like it's negative for MC channel 11 because it falls in that blue gate. What you'll find is that these gates and anything we do in the software update in real time as we move a gate. So you can see that information is being updated there for the backgating. But with these data grids, you can also apply a gate to drill down into the images that exist in that gated population. Before I do this, I'm going to duplicate this data grid. I can simply do a copy and paste command on the keyboard. I can right click on the plot and choose duplicate. Any way that you can kind of copy and paste a plot that will work in FCS Express. But what this will allow me to do is if I want to apply a gate to, say, this data grid, I can select it. And in my gating tab here, I can choose, say, gate 2. And again, those are my negatives for this NC channel 11 parameter. And if on the gate or the data grid on the right, if I want to apply a different gate, again, there's a lot of ways that you can apply gates. You can do that through the gate tab here. But additionally, if I just simply drag and drop a gate onto this data grid, it will apply it. Also, if you have your gate view or gate navigator open, if you drag and drop from there, it will apply those gates. So what these data grids are showing now are the events or the images associated with the cells in the gate. So if I move this particular gate, you can see the data grid on the left is updating to only show the events that fall within it. If I move this gate on the right, it's updating to only show the events that fall within that gate. And it's very kind of quick and easy to drill down into these populations of interest in these data grids. So there's no limit to the number of gates, there's no limit to the number of data grids that you can use, and again, you can move from plots to data grids very quickly and easily. Now, another functionality that you may want to use at some point is creating gates directly in these data grids. So you find some interesting cells by looking at the images of these, and you want to show these back on your two-dimensional plots to get some information about where these phenotypes lie, either within the same channel or within different channels. The way that we do this, again, is if you right-click on a data grid, you can choose Create Gate. And what that is going to allow you to do is check individual boxes for cells. So if I find some cells that I think are interesting, I can check these boxes, I can click OK, and what FCS Express will do is create a new gate that only includes the events for those boxes that I've checked. So what I'm going to do here is make this orange something easy for us to see. I would recommend if you're using, just a few events in a gate to also use the Emphasiz on plots feature. What this will do is if you're using a dot plot or a color dot plot, it will make the events, the dots for those events appear extra large. So what we're going to see is that we have some orange events in here. There are events that we picked. They're appearing orange in this gate. The dot size is a little bit bigger, so it's easier to visualize these. And again, if you need to edit that gate that you created, you can add or remove events from that. You're going to see it updating over in the plot once we choose Apply Changes. Right, so we can choose Apply, get those to update, or you can click OK to commit that change. So again, what we're doing here with the Amnis data is we can work with any sort of kind of plots that you want. You can create gates on these. When you have the data grids inserted, you can apply the gates over to the data grids. The data grids are very useful because you can show backgating information by visualizing that gates column. And also, if you need to drill down at the individual single cell level or few cell level to identify subpopulations of interest, you can choose that. Also, as I mentioned, if you change between different parameters, that backgating is still there. It's still going to show you all of the information for all the events that you've put in there. I should also mention that FCS Express has unlimited undo and redo. So I'm kind of pressing that undo and redo button. So if you do ever kind of make a mistake as you're performing the analysis or assigning gates, it's very quick and easy to do that. So again, this is kind of the core functionality of FCS Express to work with Amnis image cytometry data. Now, how you use these features is really up to you, but what I'm going to do is show you a few examples of what has been done in the software so far to kind of take analyzing this Amnis image cytometry data to the next level. So one example I want to show you to kind of start out with is working with cell cycle data in FCS Express from Amnis derived data sets. So I'm going to open up an example of that for you here in the software. So let me get this called up so we can take a look at it. So again, this is actually the same data set that we were just analyzing in the software. But what we've done here is we've inserted a number of plots. You can see here that on these plots, we've formatted the backgrounds to be black. Just so you see how to do this, I know it's the kind of common display that you work with in Ideas software. But what we have is a fill category here in our formatting, and you can just simply choose the make the color black, and then the backgrounds will appear black. But really importantly, when you're analyzing really just about any sort of image cytometry data that you work with, there's usually some sort of nuclear staining component to it. And the reason for that is if you need to quickly identify what is a cell. Every cell has a nuclei, at least for a lot of the biological data that we're working with here. So it's really being used almost ubiquitously for identifying a cell, and almost every experiment that you're performing with image cytometry is using some sort of DNA dye to identify a cell, segment that nuclei, go out and find cell boundaries from there, or find sub nuclear compartments. So most of the data sets you're working with have this component. Now it's really convenient because if you have this component, FCS Express has tools to analyze cell cycle data. And if you've, say, for this experiment, you've acquired DRAC5, or if you had Hosh, or if you had DAPI, essentially you have cell cycle data associated with that, whether or not you knew it. FCS Express has a tool in the software called MultiCycle DNA. Essentially, if you insert a MultiCycle DNA plot, FCS Express is going to try, or MultiCycle is going to try to automatically identify the regions. And you can see here that in this plot, we've identified, or MultiCycle has identified, the percentage of G1, the percentage of S phase, the percentage of G2. It's modeled that data on the plot just using the cell cycle, or I'm sorry, the DNA stain that happened to be there. So what I'm getting at is you have these DNA stains, and you can get information out of them that you may not have known about, right? So they don't just have to be a segmentation channel. You could use that channel to get more information from your data. So the way that we set up something like a MultiCycle plot, you can see here that we have a histogram for the data inserted. I'll do one of these from scratch so you can kind of see it. So I'm going to insert a histogram. I'm going to switch this over to our MC11, which is our DRAC5 channel. You can see also here that we've actually set up some gating akin to the gating that was done in the Ideas software, where we look at a focus gate. We have some debris gates. So I'm going to apply the region 3 gate to this plot. Now, what will happen here is, again, a lot of this data can kind of be scaled down here to the left. If we want to make this into a MultiCycle plot, you could either insert a MultiCycle DNA, or if you have that histogram, you can convert it to a MultiCycle DNA plot. But you can see here the fit is actually pretty bad. All of our data is squished down there in the left-hand corner. So again, what I'm going to do is right-click and choose Magnify Resolution. I'm just going to kind of zoom in on that area of the histogram. And you can see now that we're trying to fit this data into the MultiCycle fitting algorithm to determine the G1, the S, and the G2 populations. So what that will do is, once I have this plot, I can right click, I can show, say, DNA cycle statistics, and it's going to give me the information. Whether I'm looking at G1, S, or G2, it's going to give me percentages. And of course, with our MultiCycle DNA plots, there's an entire tab here devoted to how to kind of work with these plots and how to model it. We do have separate webinars on our YouTube channel and on our website that will cover how to work with these types of data. But importantly, that's how you insert a plot to get some of the information from this. And if you want to take that a bit further and analyze the cells within it, well, you can create some markers on a standard histogram. You can make these gates. And if I wanted to come in and say, all right, well, let's look at all the cells that are in G1, I can apply it over to our data grid. And as we move that marker, as we move that gate, everything in that data grid is going to update. So you can imagine you could have a few data grids to visualize S phase, G2, and G1. You can work with one and you can apply the gates back and forth. But again, this is kind of some free information, right? You're getting some information about cell cycle statistics from a channel that you may have just set up to identify a cell, that you may have just set up to do a subnuclear compartment segmentation and ideas software. So keep in mind these tools are here. The basic version of MultiCycle comes for free with every copy of FCS Express. So just trying this out. If you have FCS Express, it's not anything extra that you have to do or pay for in the software. So another example I want to run through with you today is an example of an immune synapse assay that was put together in Ideas software. And the idea behind this immune synapse assay was that the researcher was looking to do an IC-EC50 curve on a dose titration on some particular data. So in other software, or even if you're using Ideas, generally what you'd have to do is perform some sort of gating. You can see here in our two-page view of FCS Express, I'm going to make it back to one page that we have essentially all of our plots and all of our gating set up. We have a data grid here that has our synapse gate defined, so we're looking at the cells that are formatting these synapses. But again, what happens with this type of experiment, since we're doing some sort of dose response curve, is that we have many data files that need to be analyzed. You can see here that in our data list, we've actually loaded up multiple DAF files, and we have these set up in our FCS Express layout so that on our second page, we're performing a dose response, right? And again, we don't have to go out to Prism, we don't have to go out to Excel for this. We can essentially use the tool in our Insert tab called a Spreadsheet. And you can see here that we have a spreadsheet inserted in FCS Express. And for every data file, we've inserted a concentration. We have events that are being pulled from an actual gate in the analysis here. And what that means is that since this information in the spreadsheet is being linked directly to a gate, that as I change a gate, you're going to see that everything in that spreadsheet is going to update for us. That also means that everything in our dose response curve is going to update for us, right? So our IC50s, our line that's being plotted here, all of this information is being integrated and reported directly in FCS Express rather than having to kind of copy and paste and move out to other software tools like Excel and Prism to get a final result. Now, importantly, the way that all this works is that all of the gates have been created equally on all of the data files. So if I use my data tab and I go into this one plot, which essentially is controlling what's appearing here in this dose response curve, I can switch data on this plot to quickly visualize all the different DAF files for the information that falls within this gate. What will happen as well is if I need to break a gating position out for a particular data file, I can do what we call making this gate data file specific. So if I click on the gate and I choose data specific gate, what you will find is that as I update this gate, you can see here that our fourth point in the titration curve has been updating because it's only associated with this fourth data file. Right? So I can move the gate independently for that one sample. If I need to change to the next sample, again, I can click on the data tab, move to the next, I can make this a data specific gate. Another way that we do this is if we hold down shift while we move the gating position, it will make it a data specific gate rather than having to click the button. And I can really figure out in real time here, what's going to happen if I exclude outliers, right? What's going to happen if I change this gating position? Remember, this isn't a tool to kind of make your data better fit your gates or make your gates better fit your data, but it's a tool to help you kind of prevent having to set a gating position, copy and paste out to Prism, decide that the ICEC 50 curve isn't so great because of one of the data points, and then having to do that all over again. Again, what we get here is the ability to move these gates, see what's going on with outliers in real time, and make better decisions as a scientist with the data that we have at hand. And of course, if we need to get all of this data out to somewhere else, if we need to get it out to PowerPoint, to Photoshop, to PDF file format, essentially what we can do is go into the File tab. We can choose Export. We'll say export it as a PowerPoint presentation. I'm going to click OK, and it's going to launch this up. Let's find a place to stick this that actually exists on my desktop. I'm going to click OK. FCS Express is going to export all of these plots, all of this information out to PowerPoint. PowerPoint's going to launch up, which you're seeing right here, and we have all of the reportables for this data now in PowerPoint. The other nice thing about these plots is that these plots are high resolution, what we call vector graphics. So that means that they can be all individually resized and modified. So if this is the plot that I need, or maybe I need some sort of information about the synapse plot, I'll bring this to the front and I'll make it a little bit bigger, and I can get the information I need from this experiment. Of course, on the second page, we have our dose response curve. And again, this is a result, right? This is something you can put in the electronic lab notebook that you can work with going forward. But again, there's a lot of other ways to kind of get these results. I'm going to show you a little bit about what we call batch processing in just a moment. Before I do so, I did want to point out one other functionality in these data grids that we have. If you do have an event in these data grids or a cell that's kind of representative of the experiment and you'd like to include that in your report, what you can do is drag and drop that cell image out, and it's going to create essentially an image in FCS Express that you can place wherever it is that you want. So if you wanted to kind of show a little image of this cell over here on top or next to this synapse plot to show you what cells in this gate look like, you can simply get that into position and you can choose that as part of your report. So again, moving these images into and out of the data grid, you can just drag and drop, and again, you're going to be able to size and move them just like it would be an image or an object in PowerPoint. So moving on to another example in the software, I wanted to cover a little bit about batch processing in FCS Express. So what I mean by batch processing is if I want to get results for a series of different DAF files, you can see here that we have a similar experiment where we have a number of different DAF files. I have them arranged in our data list. But in this case, instead of working with a fully integrated report where I'm doing an ICEC 50 curve, really what I want to do is just export some information in a very high throughput manner about what's on this report for the one data file. What's our Nexin 5% positive? What's our cell death range here? What's the file name? And the way that we do this is by utilizing the data list. If we use our next and previous button, what you're going to see is that we skip from the first data file to the next data file in the data list. Everything in the report updates. Again, if we press that button again, skip from the previous data file to the next one. We can even just click on a iteration number here, essentially what is a placeholder for that data file. It's going to load that data file into the template, and we're going to complete the report for that. Now what that will allow us to do is instead of just manually clicking next and previous and saying export to PowerPoint, export to PDF via clicks in the software, we can set up in what we call our batch processing actions here. I'm going to open this up. It's in the batch tab. we can say export all the information for what's in this layout to PowerPoint. Export everything that's here to PDF. If we want to create an Excel export, we can do that as well. So I'm going to dock this window again over here, back to where it was. Um, Again, the way that this works is if I wanted to say export to PowerPoint, I can choose this. I can give it some sort of file name. I click OK. And what that's going to do is it's going to create an export to PowerPoint action in here in the batch exports. And when I click run on this batch process, it's going to export whatever's on this page to PowerPoint. So I'm going to remove this for now. The other kind of exciting capability here is exporting to Excel. What exporting to Excel will do is for every file, it will create a row. In that row, it will create columns for whatever statistics you want. So in this case, my first column is going to be for file name. So as I batch process, the first row will have the file name for sample 1 that's in the data list. The second row will have the file name for sample 2, so on and so forth until it runs out of data files in the data list. That also goes for statistics. So if I wanted to say put in the percentage of events for R4, the way that you can populate this Excel export is simply by dragging and dropping. So what it's going to do is in this kind of fifth column here, it's going to export the results for R4. And again, there's a number of ways to get information into that export. If I have all of this information in a text box already, I can simply drag and drop, and it's going to add all the information from that text box to the Excel report. So again, when you see this run, you're going to see all of this information exported. We'll actually do that now. We'll make sure that we start from the first data file. We'll click run. We'll click OK here. And what it's going to do is do 3 of 3 actions, export to PowerPoint, to PDF, and Excel. And it's going to do that across 6 iterations, or the 6 data files that we have in the data list. There's no upper limit to the number of data files that you can have in the data list. So if you wanted to run a report like this for 100, for 1,000, for 96 well plates, you can do that very quickly and easily. And what you're going to see is that FCS Express has launched up Excel. We have our file names, we have all the percentage of gated cells, all the information we asked it to export nicely and neatly collated here. What we'll also find is that in PowerPoint, we have one PowerPoint slide for every data file or every report that we created. And again, these are those high resolution vector graphics. So if you needed to grab one plot that's in here to work with this, you simply can and do it very easily. Now, one thing that you're seeing in that layout is an overlay. So I want to point this out because I know it's a functionality that folks like to work with, and I know it's kind of difficult at sometimes in ideas. So what you can see here is that when I switch to another data file, in this plot, the red overlay has updated. We'll make this a little bit bigger so you can see it. Essentially what we've done here is created multiple overlays on this plot. Now the way that we accomplish this is you simply drag and drop from the data list. So if I have a DAF file here, I want to create an overlay, I drag and drop, say add the file to a plot as a new overlay, and that overlay gets created. And you can do this with as many overlays as you want. It doesn't have to be one data file at a time. I can drag a few in here. It's going to create overlays on that plot. Now the trick is here to format these so they look a little bit nicer, right? These overlays that I've inserted, of course, don't look like the red and black ones that I have there. So what we do is we double click on that plot. I'll get some of these other windows out of the way so it's a little bit easier to visualize. But in the overlays category, what we can do is select all the overlays that we have, and if we want to smooth them, we can apply a smoothing. Right? If we want to fill the histogram area for all these, we can fill it. Right? If we want to provide some sort of transparency for these, we can do so. And when you do this, you can really accomplish and create any sort of visualization of these overlays and of the data that you want. You're going to get real-time displays of what these changes will do when you're working in our formatting dialog. So again, if you don't want to fill it, take the fill out, right? There's a lot of different kind of options here that you can do. But again, you end up with this high resolution graphic that was quickly and easily created. Now, the last thing I want to leave you with today, before we start wrapping up, is working with TISNI data or working with TISNI algorithms for Amnis data. So this is pretty cool. One of the benefits of working with Amnis instruments is that you get very highly multi-parametric data sets. You get intensity parameters, you get shape parameters, area parameters, all sorts of descriptors of cells. The problem becomes is that when you have this data, there's so many parameters that sometimes it can be hard to figure out what's there and make some decisions about this data. But there's tools out there like TISNI and VISNI and SPADE, various clustering tools. They kind of help assist researchers to explore the data, to make some decisions. And many of these tools are in FCS Express. The way that we access them is through the Tools tab. We choose transformations, and all the transformations that are available will be accessible by dropping down this little dialog here. And if we want to add a TISNI transformation, you can pick that. And then the dialog for working with TISNI pops up. It's a very easy to use interface where essentially we select the parameters we want to use in the algorithm. We select some sort of downsampling, some of the variables associated with this. And once we have that TISNI created, you can essentially take it and apply it to a plot just by dragging and dropping onto a plot that's going to create that TISNI parameter, the TISNI X and Y parameters. It's going to calculate it and it will update it on the plot. Now, of course, we could talk about TISNI with any sort of data sets for hours. We do have separate resources and webinars on our website and on our YouTube channel to discuss this. But what I want to show you is just simply how that works with the image cytometry data. So if I wanted to insert a data grid, of course you can still insert a data grid. You probably want to get your TISNI transformation, or you do want to get that applied to that data grid so you can visualize what's there. What we've done here is actually three transformations. One is we've only looked at intensity parameters in the clustering. Another, we've only looked at morphometric area parameters with the subset. And the last one here, we looked at morphometric and intensity parameters. So this is the one I'm going to be focusing on with our data grid. But what you'll find is that if I take a gate and I apply that over to our data grid, as long as I have that transformation applied to the data grid, we're going to visualize the events that fall within these TISNI clusters. So you can see here that I have this plot. It's set to show the heat based off of channel 2 in the data set. This is showing the heat based off of channel 3. This is showing the heat based off of channel 4. And we can see that when we look at this really hot population here, of course, in the channel 2 Alexa Flora 488 channel, these are all positives. Now if I move this gate around and I try and grab some cells that are appearing over here in the channel 3, but not in channel 2, you can see that they're highlighting over here in the data grid, right? And again, if I kind of grab some stuff that's hot over here for channel 4, you can see that it's moving that gate on channel 4. Of course, they're hot in the data grid, and they're cool for the other channels because the other channels aren't really clustering into there. So again, you can work with these tools with Amnis data. You can get the direct linking back to the images. So really any of the kind of clustering, any of the kind of TISNI, VISNI, SPADE analysis you might be doing with flow cytometry data, it's directly applicable for working with Amnis data files in FCS Express. And again, you get that direct linking to the images, which is one of the most important things I think about this. So with that, I'm going to kind of summarize a little bit about what we've learned today on the webinar. Importantly, Amnis Data and FCS Express can be used like any other flow cytometry data, but you maintain access to your raw images. You don't have to export from IDEA software out to FCS files. You can use the native DAF and SIF files that are there. What that will allow you to do is skip many of the hurdles for presenting and reporting your Amnis data that you have in IDEA software. So if you need to simply create an overlay, if you need to get a figure ready for publication, All those things are very quick and easy to do in the software. Additionally, if you need to kind of get into higher throughput, if you need to work with batching, if you need to do things like working with regression curves, if you need bar charts, pie charts, if you need to work with cell cycle data, all of these tools are directly integrated into FCS Express. So it's kind of a one stop shop for a lot of the things you might be doing in multiple software packages currently. Also, it's important to note that you can easily perform cluster analysis, you can easily perform t-SNE and other transformations, all while directly accessing those images or retaining that access to your raw images at any time. So with that, we're going to stop. We're going to take some questions. I want to say thanks to everybody who came out for the webinar today. We really appreciate it. If you have any questions after the webinar that we didn't get a chance to address, please contact us at support@denovosoftware.com. You can also visit us on our website at denovosoftware.com. If you currently have FCS Express, there's a lot of information there for you. If you don't have FCS Express, you can download a free demo of the software and you can try it out. But again, thanks for joining us, and we'll stop for some questions. We hope everybody has a good day. Bye bye.